Chapter 3, it's about cytology or study of the cells. So the study of the cells is important because all organisms are composed of cells, which are the structure and functional unit of a living organism. When you study the cells in an organism, you can be able to know the location, the shape, the function of the cells, and how they integrate their functions into the complete living organism or the human body. Also, when you study the cells, you can find what are the mechanisms of disease and provide a rationale for therapy. We have millions and millions of cells in our body, and they are classified as 200 types. And these 200 types of cells, they can have varied shapes. And here are some of the most common shapes that you can find. Like if your cells are thin, flat, or scaly, they are classified as squamous cells. If the cells have a shape like a square, they will be called cuboidal. If the cells are taller than wider, they will be columnar. If the cells have multiple angles and multiple size, they will be called polygonal. If the cells have the shape like a star, they will be called stellate. If the cells look round to oval, they will be called spheroid or ovoid. If the cells have a disc shape, they will be called discoid. If the cells look like a spindle, where they have a thick portion in the middle and taper ends, this will be called fusiform cells. And if the cells are elongated, looking like a thread, they will be called fibrous. Now, the shape can appear different when you view the cells at a longitudinal section versus a cross section. So, in these cells, we are looking at longitudinal sections. So here is how the squamous cells look like. They're flat or thin. How these cuboidal cells look like a square. How the columnar cells look like a column. How the polygonal looks like. They don't have a definite shape. Stellate cells look like a star. Spheroidal cells like a sphere. Discoid shape like a disc. Fusiform shape like a spindle with a thick center and then taper ends, and then fibrous like a thread. Now, usually when you have a figure showing the cells, it will have a nucleus shown inside the cells. So this will be the nucleus of the cells, and in the case of the spheroidal cells, this will be the nucleus. The cells, they have a shape, not only a shape, but also a size. And the size vary according to the cell. The range of the diameter or size of the cells can go from 10 to 15 micrometers in diameter, with the X cells being large cells that can measure up to 100 micrometers in diameter, and longer cells like the nerve cells, they can measure up to 1 meter in length. Now, the cells can have a limit on their cell size because the cells that are very large can burst. And there is this formula that helps us to calculate how much volume the cell can have based on the increase in diameter. Now, the volume is proportional to the cube of the diameter, and the surface area is proportional to the square of the diameter. So here is the effect of cell growth in volume. So if you have a 10 by 10 cell converted into a 20 by 20 cells, then this is how you change the volume. So there is these factors where it tells you that the diameter is increased by a factor of 2, and the surface area is increased by a factor of 4. So it's the square of the diameter, and the volume is increased by a factor of 8 because it is the cubic exponential of the diameter. So basically 2 by 2 it's 4 for the surface area, and 2 by 2, which is 4 by 2, will be 8 for the volume. And you can see it in here. So the diameter is 10. If you multiply it times 2, the factor 2, that will give you 20. For the surface area, you have to multiply it by a factor of 4. So 600 times 4 will be 2400. And then 1000, which is this, the volume, can be increased by a factor of 8. So 1000 will give you a volume, if you increase just the diameter, for a factor of 2 of 8,000 micrometers that are cubic micrometers. Now the cell has basic components that can be studied with different microscopes. The microscopes that we use in the lab, that is most commonly used in education, is a light microscope. With the light microscope, you can see structures like the wrapping of the cells, which is the plasma membrane. You can see the nucleus, and then you can see the cytoplasm, which is the fluid that it is located between the nucleus and the plasma membrane. We have more sophisticated microscopes that give you more detail, like transmission electron microscope, and it gives you sometimes an atomic resolution of the structures of the cell. And then you have a scanning electron microscope that it is also very detailed in their views, and it can give you a three-dimensional shape of the internal structures of the cell. Both of these microscopes, transmission and scanning electron microscopes, can be used for research purposes. So this is just to compare how does this cell, which is called a eosinophil, look with a light microscope on the right side, and how it will look with a scanning electron microscope, which is on the left. So this is the nucleus of the cells, and all of these that you see here, which are named as lysosomes in here, these are the little granules that you see in here. So what are the basic components of a cell? So they have a wrapping, which is called the plasma membrane or cell membrane. So this plasma membrane sets the limits or boundaries for whatever is inside of the cell and outside of the cell. The plasma membrane is made out of phospholipids, proteins, and cholesterol. The cytoplasm will be a fluid that it is located inside the cell, and within this fluid you have several components of the cells, like organelles or little organs, cytoskeleton, which is made out of proteins that gives this structural support to the cells and also gives the shape to the cells. And also you can have several spheroid look-like structures that are called inclusions. Within these inclusions you can store nutrients or foreign particles or enzymes. And then you have within the cytoplasm the cytosol, which is actually the fluid portion of the cytoplasm that is also known as intracellular fluid or ICF. Now the plasma membrane, as we said, set a boundary for the cells. So any fluid outside of the plasma membrane is going to be called extracellular fluid, or also known as tissue fluid or interstitial fluid. So this is the structure of a representative cell, in which you have here the plasma membrane, you have the cytoplasm with its organelles, and then you have the nucleus in here. Now, the cells, when it comes to a particular tissue, which is known as the epithelial cells, they have different sides. So they have a side that faces the environment that is called the apical side, and they have a side that faces a structure that is called the basement membrane. This will be the basal side, and then it has two lateral sides that faces two other cells, neighboring cells. And the cells are usually in the epithelial cells hold into structure, so they won't be floating. And these connections are known as cell junctions. So here is one example of a cell junction. This is known as desmosome next to the apical surface. And then in here you have the hemidesmosomes next to the basal surface. And some of these organelles in here, besides the nucleus, are the following. So this is the rough endoplasmic reticulum in here. This is the smooth endoplasmic reticulum. These are mitochondria. This is the Golgi complex or Golgi apparatus. Then you have the Golgi vesicles. Then you have secretory vesicles that are going to deliver materials into the outer part of the cell. And then you have centrioles, which are this. You have this cytoskeleton made out of 3 different types of proteins. And of course, you have these connections to the cells. So this is a 3-dimensional view of the basic components of the cell. So again, this is the nucleus. This is the rough endoplasmic reticulum. This is the smooth endoplasmic reticulum. This is the Golgi complex. This is the mitochondrion. This is the centrioles. This is the inclusions or vesicles. And this will be microvilli and cilia. Now, not all the cells have a cilia, and not all the cells have microvilli. So if the cells have cilia, they don't have microvilli. So let's talk about the details of the plasma membrane. The plasma membrane, it is the boundary of the cell. And when you look at it under the microscope, under the electron microscope, it looks like dark parallel lines, 2 lines. And then this plasma membrane, it has these faces, one that goes and it's located towards the cytoplasm, which is called the intracellular phase, and one that is oriented away from the cytoplasm. This will be the extracellular phase. What are the functions of the plasma membrane? To define the cell boundaries, to govern the interactions with other cells, and to control the entry or exit of materials to and from the cells. So this is a view of the plasma membrane with the electron microscope. So here is one of the components of the plasma membrane. This is the intracellular phase, and this will be the extracellular phase. And this part in here is the nuclear envelope which has similar appearance to the plasma membrane. And this is the nucleus, and this is the cytoplasm. So if you see this cartoon of the plasma membrane, you can see some of the components. So the plasma membrane, the majority of the plasma membrane is made out of these phospholipids, which have 2 fatty acids connected to a phosphate group. The fatty acids, as we saw in the previous chapter, they are hydrophobic because these are lipids. And then the phosphate group is hydrophilic. So the plasma membrane is made out of 2 rows of phospholipids. One that faces the environment, which will be located towards the extracellular fluid, and one that faces the cytoplasm, which will be this. So the plasma membrane is a phospholipid bilayer. And then within this plasma membrane, you have molecules of cholesterol that I already told you in the previous chapter, that cholesterol prevents the plasma membrane from being overly fluid because phospholipids are very flexible, and you impart some rigidity by having some molecules of cholesterol. Now, embedded within this phospholipid bilayer of the plasma membrane, you have proteins. And these proteins can be channels like this one in here, which is like a little gate or door that allows substances to go in and out of the cells, or they can be transmembrane proteins. These transmembrane proteins, what they do is they can send signals from the environment, and then these signals can be transduced through these peripheral proteins, or can be given by these peripheral proteins into the internal components of the cells, so that the cells can change its behavior according to whatever signal this transmembrane protein is receiving. So these transmembrane proteins can be seen as receptors. So for instance, if a cell needs to grow, it will have receptors for growth hormone. And this growth hormone will bind into this transmembrane protein, and then this transmembrane protein is going to sense the presence of the growth hormone, is going to send a signal through this peripheral protein, and the cell will start making more proteins so it can grow. Now, some of these proteins will be conjugated or be having chemical bonds with glucose, like in this case, glycoprotein, or even the lipids. They can be combined or conjugated with glucose, like in this case, these glycolipids. And all of these glycolipids, glycoproteins, which are either proteins combined with carbohydrate chains or lipids combined with carbohydrate chains, they help to receive signals or to attach to other cells. Now, underneath the intracellular portion or face of the plasma membrane, you have this support that it is made by proteins of the cytoskeleton, like a fibrous protein that is called actin. So you have extra support just beneath the intracellular face of the plasma membrane. And as you can see here, this is the intracellular fluid. So this is the internal part of the cell, and this is the outside of the cell. Now, the plasma membrane is made out of 98% of lipids, and out of those lipids, 75% are phospholipids. And as I said before, these phospholipids are arranged in a bilayer, so you have two layers. And these phospholipids are amphipathic, so they have a phosphate group that is hydrophilic, that can interact with water, and then the tails of the phospholipids, which are the fatty acids, are hydrophobic. So hydro means water, phobos means fear for hydrophobic. For hydrophilic, hydros means water, philic means love, so molecules that love water. And then these phospholipids are not static. They are drifting laterally and even rotating, keeping the membrane fluid. Now, 20% of the plasma membrane lipids is made out of cholesterol that helps to hold the phospholipids still and can stiffen the plasma membrane. 5% of the membrane lipids are combined with carbohydrate chains, so we have glycolipids. And some of them, they have more carbohydrate chains than lipids or even proteins, and they form like a thick membrane they form like a thick layer outside of the membrane that is called the glycocalyx. This glycocalyx is like a sticky surface that helps the cells of the connective tissue to hold into the surrounding kind of almost empty space. But of course, we don't have an empty space within our body. But at some point, that's the function of the glycocalyx, to stick the cells to the surrounding area so that the cells won't be floating. Now let's talk about the details of the membrane proteins. So the membrane proteins make only 2% of the plasma membrane molecules, but they make 50% of the weight of the membrane. Why? Because the proteins have a greater molecular weight than the lipids. Now we have a major classification of these proteins. So we have integral proteins that penetrate completely the plasma membrane, or are also known as transmembrane proteins. And then we have peripheral proteins that only adhere to one face of the membrane, but do not penetrate it like the integral proteins. And these peripheral proteins are found within the inside of the cells and are tethered to the cytoskeleton. Now, just similarly as for the lipids, the integral proteins have a hydrophilic region that it is in contact with the cytoplasm and the extracellular fluid, and they have a hydrophobic region that it is located within the lipids of the plasma membrane. And similarly as the phospholipids, they are not static. They are drifting in the membrane, and others are anchored to the cytoskeleton. So this is this cartoon showing you how the transmembrane proteins look like. So this will be the hydrophilic portion of this integral protein, and it has only one polymer of proteins. In this part of the hydrophilic portion, which is outside of the cells, it is combined to this carbohydrate. So this is a glycoprotein, and it has this region of the protein which is hydrophobic, because it's located here within the phospholipid bilayer. It has this other portion which is hydrophilic that binds to the cytoskeletal protein, or an anchoring peripheral protein, which is this. And this is a transmembrane protein with multiple passes. So this is a single pass integral protein, and this one has several passes. So this one has only one, and this one has 1, 2, and 3. So this is larger than this. So again, the functions of the membrane proteins will be as receptors and also as second messenger systems. Once they receive the signal, they send it inside into the cells. And many of the proteins, the globular proteins, are enzymes. These are catalysts, again, that we saw in chapter 2. So this catalyst increases the speed of reactions, the chemical reactions. Some of them, they are gates or channels. Some of them are carriers. Some of them are found in particular cells, making an identity marker for the cells. And some of them, they are anchoring proteins that hold the cells into the neighboring cells. So these will be cell adhesion molecules, or into the environment, not necessarily into other cells. So this is the figure to show you the different functions of the membrane proteins. So this is a receptor that has a place to bind ligands, like these chemical messengers. Some of them can be enzymes that actually work at the plasma membrane. So once they bind a ligand, they destroy the ligand. And some of them, they are channels in which you allow the passages of ions. And some channels are open, so they constantly allow the pass of electrolytes, for instance, and some of them, they are gated, so they can open and close, allowing selectively passage or selective passage of molecules at a given time. And also, they will not allow anything to come in, only what it is specific for this channel. So we have, for instance, sodium channels, we have potassium channels, calcium channels that only allow the passage of these ions. And then we can have these cell identity markers. For instance, in lymphocytes, we have CD8, which is an identity marker. It's a protein. And then we have the cell adhesion molecules that helps to bind cells one to another or to the environment. So receptors bind chemical signals, and they can generate these signals that can be amplified by second messenger systems. And the enzymes, they not only catalyze reactions that can digest molecules, but they also can produce these second messengers. There is this enzyme that is called adenylate cyclase. So this adenylate cyclase can produce cyclic AMP or adenosine monophosphate, cyclic adenosine monophosphate. So this will be a second messenger. Now, channel proteins will allow the passage of hydrophilic solutes and water to pass through the membrane. For instance, we have channels that are specific for the passage of water that are called aquaporins. And like I said before, some of them are always open and some of them are gated. And out of the ones that are gated, we have something that is called ligand-gated channels, which only open when you have a chemical messenger bound to these channels. It's like if you have a lock and key mechanism. So you insert the key in the lock, so you have a physical connection, and then you open your door. And some of these channel proteins are voltage-gated channels that respond to charge changes or voltage changes. It's similar to what happens when you have a key card. So in the key card, if you want to open a door, you don't have to have any physical interaction between the key and the lock. So it's a change in voltage. It's like as well for opening the doors on a car, that it is a keyless entry door. And then you have mechanically gated channels that respond to physical stress on the cell. So if you have pressure on the plasma membrane or some physical distortion, then these channels open. And the channels are very crucial for the function of nerve and muscle cells, because through this function of allowing electrolytes to come in and out, these cells, excitable cells, the nerve cells and the muscle cells, can function. Another function of proteins is to be carriers, in which they bind solutes, particles that need to be moving across the cells. Some carriers are pumps that they use energy, they use ATP. And as I said before, some proteins are cell identity markers. Some molecules are cell adhesion molecules, some proteins, and they link the cells to the extracellular material. Now, examples of second messengers, it's the conversion of, the actually binding of epinephrine to the cell surface receptors for epinephrine, and this epinephrine will bind to a protein in the plasma membrane that is called G protein. this G protein, it's an intracellular peripheral protein that will get the energy from GTP. That's why it's called G protein. And when the epinephrine binds to this G protein is going to send a signal to this adenylate cyclase, and it's going to convert ATP into cyclic AMP, which is the second messenger. And then this cyclic AMP is going to activate enzymes inside the cells that are called kinases. And these kinases are going to phosphorylate or add phosphate groups to other enzymes to amplify the signal. And then your cell can respond to that amplified signal. It's like when someone goes to the door, knocks your door, and a little children answers the door and is asking for an adult. And that children knows that the adult is kind of far away from the door and starts shouting. So it's amplifying the signal, just to let know that someone is there at the door. Now, these G proteins are very important because 60% of the drugs that are used in medicine works through the action or modifying the action of G proteins as second messengers. So this is how the second messenger system works. So you have, in this case, epinephrine as a red triangle that binds to this receptor for epinephrine. And this G protein, which is attached to this receptor, is going to migrate from the receptor into this other integral protein, which is adenylate cyclase, which is an enzyme. This adenylate cyclase, once it's bound to this G protein, is going to convert ATP into cyclic AMP, which is the second messenger, and to 2 molecules of inorganic phosphate. And then this cyclic AMP is going to activate phosphorylators like an inactive kinase, and this inactive kinase will become activated and is going to phosphorylate other enzymes or other molecules inside the cells, so you can have various metabolic effects. Now, the glycocalyx is a layer that is outside of the cells, made out of carbohydrates and proteins, more carbohydrates than proteins. And it's very sticky because carbohydrates itself, they are very sticky. And they form a very disorganized structure that it looks like a fuzzy coat, and it is unique in everyone but in identical twins. Some functions of the glycocalyx will be to protect the cells, to provide immunity to infection, to defend against cancer, to be able to identify persons who are compatible for transplant, to adhere the cells for fertilization, and for the development of the embryo. Now, another structure in the cells is called the microvilli. The microvilli are extensions of the plasma membrane that measure between 1 to 2 micrometers in diameter, and they give this increase of surface area in the plasma membrane. So you can have a greater surface area of 15 to 40 times. And this microvilli, it is found in cells that needs to absorb a lot of substances. So in some cells, like the small intestinal cells in the jejunum and the ileum, this microvilli looks like a brush border. And inside this microvilli have actin filaments that gives certain structure so these microvilli don't get squished. And they have some blood vessels that helps to absorb the nutrients that goes across this microvilli. So this is how the microvilli will look like. They are extensions, but they are short, much shorter than the cilia, which are not just colored with green color. So the microvilli here are colored with green, and the cilia are not colored in this case. If you make a longitudinal section, like what you see here in this part A or panel A of the figure, you will be able to see the internal structures of the microvilli. So these are actin filaments that provides certain structure, so they don't get squished as easy, so they maintain certain position. Now this microscopic view is done with a scanning electron microscope, and these colors that you see here has been enhanced with the computer. Now, in order for us to see cells, we have to prepare the cells, so we have to make a preparation of the cells. Depending on what microscope are you going to be using is going to be the technique for staining the cells. But one of the simple methods of staining the cells is hematoxylin and eosin staining technique, which you just add these two stains or dyes into the sample of cells over a glass slide. Then you rinse this excess of dye, you put a cover slip or like a cover glass on the top of the glass slide, and then your internal structures of the cells will be visible. Unless you stain the cells with a special technique, you won't be able to see the cells. Now, in here on panel B, you can see a cross-section of microvilli. And the microvilli here in the center is showing these actin microfilaments that you see here on the longitudinal view. And this will be the outer edge of this microvilli. Now let's talk about the other structure of the cells, the cilia. So cilia are hair-like processes that are long, between 7 to 10 micrometers in length. They are single, non-motile primary cilium that are found nearly in every cell, and they help to monitor the conditions of the environment. For instance, we have this cilia in the inner ear that detects the motion of your body so you can maintain balance. Also, we have cells that has multiple non-motile cilia, like the ones that we have in the nose, which are sensory cells. And some cells, they have only a single cilia that it is motile, like in the case of the cilia that it is found in the sperm cells. Or we have cilia as well that is motile and are multiple, like the ones that we have in the epithelium or the covering of the respiratory tract, the uterine tubes, the ventricles of the brain, and the ductus of the testes. And the function of these cilia in these areas that I mentioned is to transfer, for instance, in the respiratory tract, the mucus, so we can sweep away things that comes into our respiratory tract that can damage our lungs. In the case of the uterine tubes, the cilia will transport the egg so that the egg can be fertilized, or the fertilized egg can go into the uterus, into the lumen or into the cavity of the uterus. And in the case of the testes, the ducts of the testes, they help to transport the sperm cells. Now, the cilia has this motion, which looks like waves of sweeping waves that helps to move the materials across the surface in one direction. And they have these power strokes that are followed by recovery strokes to propel the substances much faster. Now, some cilia might have defects in their function because the structure is damaged, and then you will have a disease that you will have a specific name. But as a general name, anything that damages the cilia or that it prevents the function of the cilia is called ciliopathy. Now, inside the cilia, you have a core that is called the axoneme. And within the axoneme, you have microtubules, which are cylindrical structures, hollow structures, that has this protein that is called tubulin. And there is this arrangement inside the cilia that forms like a little cylinder that it is arranged in a 9+2 arrangement. So you have two central microtubules surrounded by a ring of 9 pairs of microtubules. Now this ring of 9 pairs will anchor the cilium to the cell as part of the basal body. And then within this cilia, you have a protein that is called dynein that has like a little arms that crawl up an adjacent microtubule bending the cilium. And this motion of the cilia uses energy from ATP. Now there are some diseases that affects dynein, that protein, and the cilia won't be motile. So it won't move, and the person will trap mucus within several areas where this cilia is present. So if you take a structure like a cilia and you make a cross section on the shaft of the cilia, you will be able to see the arrangement of the microtubules. So this will be the core, and you have a 2 to 9 arrangement. So you have 2 microtubules in the center and 9 pairs of microtubules around this, and this is called an axoneme. And in here, connecting the microtubules, you have this protein that is called dynein, and this is a transmission electron microscope with enhancement of these structures, color enhancement, so you can see the 9:2 arrangement of the microtubules. And then with a scanning electron microscope, you will be able to see some of the epithelium or the coverings in our respiratory tract that has cilia that looks like a shaggy carpet. Now the cilia is capable of beating freely within a saline solution, and it's because within the cilia you have chloride pumps that pumps chloride into the extracellular fluid. And when you pump chloride, sodium inside the cells and water will follow, because chloride, it is negatively charged, and sodium, which is positively charged, will follow the negatively charged chloride, and the water will follow sodium. Now mucus will float then, because of this, on top of saline solution. So this is the figure showing you the ciliary action. So outside of the cilia, when you have this saline solution, you have this beating action of the cilia in one direction, sweeping away excess of mucus on the top of the cilia in the respiratory tract. So if the cilia move in the direction where they're beating out the mucus, that will be called a power stroke, and then they can have a less potent stroke that is called a recovery stroke. So one of these ciliopathies or diseases of the cilia is known as cystic fibrosis. This is a hereditary disease in which the cells make these chloride pumps, but they fail to put them in the plasma membrane of the cilia. And then the chloride pumps, they cannot make a natural saline solution that will go and top the cell surface, and then mucus will accumulate within the respiratory tract, within the reproductive tract, and within the pancreas. Now, when a person has cystic fibrosis, it will have high chances of dying early in life, like around its 30s, because the mucus that is accumulated in the respiratory tract will be very thick, and it will prevent the air from coming into the lungs. So the person will have hypoxia or low absorption of oxygen. And not only that, but the person will have chances of having chronic respiratory infections, which ultimately will be one of the complications of cystic fibrosis and the cause of death. I guess, sorry, I mentioned that sperm cells have cilia, but no, they don't have. They have a flagella, which is longer in extension than the cilia. And this is actually the only cell in the body that has flagella. And this flagella is motile, similarly as for the cilia, but the flagella in the sperm cells is much thicker, and it has a more powerful stroke than the one for the cilia. And this flagella will move in a zigzag motion or a whip-like motion, allowing the transportation of the sperm cells in a rapid way. Another extension of the cells or structure of the cells, it's pseudopods. Pseudopods is false feet. Pods stands for feet, pseudo for false. So these are changing extensions of the plasma membrane that can vary in shape and size according to what is the need of the cells. Pseudopods are found in cells that need to move from one place into another. So they are used for locomotion and in order for the cells to capture foreign particles. One cell in our body that it is rich in pseudopods are the neutrophils or the macrophages. So this figure, it shows you pseudopods in this cell to trap bacteria. And these pseudopods can be short or they can be long, as in this case. Now, let's talk about membrane transport. So the plasma membrane, which is a barrier of the cell, allows certain substances to come in and out, and it is selectively permeable depending on what are the needs of the cells and what is the type of molecule that needs to come in and out. So a membrane transport that doesn't require ATP is known as a passive transport because it doesn't require energy. And a membrane transport that requires ATP is known as an active transport. Now, examples of passive transport will be filtration, diffusion, and osmosis. And this transport happens because you have a difference in concentration of the particles. So the particles will tend to move from an area of high concentration into an area of low concentration. So from more to less until you reach an equilibrium. And this movement or passive transport happens because within these molecules you have random molecular motion, and these particles will collide, and this provides the necessary energy for the motion of the particles so that they can be transported. Now, in active mechanism, you have these pumps that allows you to move these particles against their concentration gradient. So you can move things from less concentration to more concentration, from less to more against their concentration gradient. So you are forcing molecules to go across the plasma membrane. Now, examples of active transport will be active transport itself and vesicular transport. And also another form of cell transport will be carrier-mediated transport, in which you have a membrane protein that binds to substances and move them across the plasma membrane. So this is called facilitated diffusion. So now let's talk about the specific modes of transport. So for passive transport, the first one, filtration. So the particles in this case will move through a membrane driven by physical pressure. Examples will be filtration of water and small solutes through gaps that we have in blood vessels that are called capillaries. So these capillaries, they have pores, and then water or solutes like electrolytes or amino acids can move across these capillaries. So through this filtration process, you can deliver water and nutrients to the cells and also remove waste from the capillaries in the kidneys. So this is a diagram or a figure showing you how this filtration process happens. So you will have a pressure inside this capillary, this blood vessel, that allows the exit of water and solutes. These solutes can be nutrients like glucose, it can be amino acids, and also they can be gases like oxygen. So see how you have pores here within these capillaries. Now in simple diffusion, which is also a passive mechanism of transport, you have the movement of particles from an area of high concentration to low concentration. And the movement of the particles will happen because the particles are in constant motion. And as these particles are moving, they will collide and they will bounce off each other, and then they can be kicked out from an area of high concentration into an area of low concentration. Now, some things that can affect simple diffusion will be how concentrated is the particle. So the more concentrated the particle is on one side than in the other side of the membrane, the more diffusion that you will have. Also will be how much plasma membrane you have available. If you have very little plasma membrane, the diffusion is going to slow down. Another important aspect is that affects simple diffusion is temperature. When you increase the temperature, you have more random motion of the molecules. The molecules tend to collide the most and they tend to diffuse the most. So simple diffusion again, the substances will diffuse down their concentration gradient, so from more to less. And you don't necessarily require a membrane, but if you still have a membrane, the substances will move from more to less. And then the substance will stop diffusing when you reach an equilibrium. So when you have equal number of particles on one side of the membrane and also in the other side. I already mentioned what it affects simple diffusion, so temperature, also molecular weight. I forgot to tell you this. So the heavier the molecule, the less simple diffusion you will have. Because, of course, the larger molecules will tend to have more resistance to motion because they are heavier. And then the increased difference of gradient or steepness of the concentration, the greater the rate. The greater the surface area, the greater the rate of diffusion, the greater the permeability of the plasma membrane or the membrane, the greater the rate of diffusion as well. Now, osmosis is diffusion of water through a selectively permeable membrane. And water will move from an area where you have more water to an area where you don't have as much water. And if you have particles diluted within these sides of the membrane, diluted in water, some of these particles can draw water to the other side. And this is crucial for considering what type of IV fluids can be introduced into the person's body. So one of the reasons of why people have diarrhea is because sometimes when they have a damage in their epithelial cells of the intestines, these solutes in the foods that they ingest, they cannot be absorbed. So these solutes like glucose can be accumulated within the internal opening of the intestines, which is called the lumen, and then the water, in attempt to decrease the concentration of glucose, will follow this glucose, and then you will have diarrhea. Also, this is the basis for the opposite constipation and edema. Now, water will diffuse through the phospholipid bilayers using these aquaporins, or channels specifically used for water. And then the cells can speed osmosis by putting more aquaporins under the influence of some hormones, like in the case of antidiuretic hormone. So this is a figure to show you the concept of osmosis. So you have in this shared container a site A and a site B separated by this semi-permeable membrane. In this semi-permeable membrane, only water can move across. And in the case of site B, you have two solutes. So you have this solute with these purple spheres that you can see, and then you have this big solute with this orange color. So because of this, water will tend to flow from site A to site B to decrease the osmolarity of this compartment of site B, the concentration of solids within site B. And then if you let it go, you will reach an equilibrium 30 minutes later. So site B will increase its volume while site A will become smaller because you have loose volume. Now at some point you reach the equilibrium when the osmotic pressure equals the hydrostatic pressure. So the osmotic pressure will be the pressure that makes water to flow from compartment A to B, and hydrostatic pressure will be the pressure that opposes the osmotic pressure, preventing water from moving more into site B. So osmotic pressure is the hydrostatic pressure required to stop osmosis, and increases as the amount of non-permeating solid rises. And reverse osmosis, it's a mechanical procedure that helps to override osmotic pressure, and this will help to keep solids on certain areas where you don't want them. Like in the case when you're purifying water, you can use reverse osmosis to take away any contaminants from the water. One measure of osmolarity, or the concentration of solids within a solution, one unit is called osmoles. So 1 osmole is equal to 1 mole of dissolved particles. And this is taking into account when you want to have a solute to ionize in water. So in the case of glucose, 1 molar glucose, this is 1 osmole per liter. And when it comes to sodium chloride, is 2 osmoles per liter. Now osmolarity will be the measurement of how much or how many osmoles you have, or solids you have, in a liter of water or a solution. Now our body has many solids within the fluids, and this will contribute to the blood osmolarity. And this blood osmolarity will be the combination of the different osmolarities provided by the different solutes. How much osmolarity we have is 300 milliosmoles per liter. So it's 1,000 of us osmol. And what are the three substances or three things that contribute to the osmolarity? Will be glucose, will be sodium, and chloride. These three can contribute the most. Other solids can also contribute, but these will be the ones that contribute the most. And it's basically how much concentration of this you have in your blood. Now, tonicity is the ability of a surrounding solution or a bath to affect the fluid volume and pressure in a cell. So this tonicity will depend on the concentration of the solutes that don't move across the plasma membrane. So if you measure a solution for its tonicity, you can have two types of solutions. So you can have a hypotonic solution, which will cause the cells to absorb water and swell because you have low concentration of solutes with regards to the solvent that are found within the solution. And this solution, if you put it in the cells, in the body of the person, this hypotonic solution will make the cells to absorb and to burst eventually. Now, in hypertonic solutions, you will have a high concentration of solutes in the solution, and this concentration of solutes is greater than the solution or the osmolarity that you have in the intracellular fluid. So this hypertonic solution, if you put it within the cells will lose water because the water will move towards this hypertonic solution to decrease its concentration of solutes, and the cells will shrink or crenate. Now, if you have an isotonic solution, the solution will have the same number of solutes in the solution as what you have in the intracellular fluid. So no water movement will happen from the cells, so the cells will retain their volume. An example of isotonic solution will be a normal saline solution. So it has a concentration of weight per volume of 0.9% of sodium chloride. So this is the effects of the tonicity of different solutions of red blood cells. So if you put a red blood cell in a hypotonic solution in which you have low concentration of solutes within this solution, the water inside these red blood cells will be very few, and you will have increased concentration of solutes inside the red blood cell. So the water will move from the hypotonic solution into the inside of the cells, and the cells will burst as they're gaining volume. So you will have hemolysis. Now, in isotonic solution, you have the same concentration of solutes in this solution that it is outside this red blood cell, and you have the same concentration of solutes inside this red blood cell, like the 0.9 sodium chloride solution, so the cells will stay at the same volume. So it won't gain volumes, but it will not decrease its volume. Now, if you put a red blood cell in a hypertonic solution in which you have a lot of solutes, this cell will start losing water because water will follow the solutes that are found within this hypertonic solution. So the cell will crenate or shrink or dehydrate. So one way that I use to illustrate osmolarity in our blood, guys, is whenever you ingest ice cream, which has high concentration of glucose, or whenever you ingest popcorn in the movies where they put a lot of salt, or French fries where they also put a lot of salt in it, you become more hyperosmolar, so you gain a lot of solutes. So water and glucose increases our osmolarity. And because of that, you become thirsty because your cells will be in a hypertonic solution, so they will crenate, they will shrink, they will dehydrate. Because the water from your cells will go into your bloodstream trying to dilute the excess of sodium or the excess of glucose that you have in your bloodstream. Every time that you eat ice cream or hot dogs, which are also so salty, pizza, French fries, or when you drink highly sugared drink, like if you add too much sugar into your coffee or into your tea, you dehydrate yourself because you become more sugary. You have hypertonic solution in your blood, so you increase your osmolarity. And your cells will give up water just to decrease this osmolarity. And you start your thirst mechanism so you can compensate. So you can drink more water so you don't have to lose as much water from your cells. So hopefully this helped you to clear up the concept of osmolarity and tonicity. Now for carrier-mediated transport. So this carrier-mediated transport is the process of carrying substances across the plasma membrane by the use of proteins that are located within the plasma membrane. And these solutes that you are carrying, or these molecules, they can be carried into the cells or out of the cells, or into an organelle or out of this organelle. Now, the carrier-mediated transport is specific and it can be saturated. Why? Because you can only have a certain number of transport proteins within the plasma membrane. And if you have more solutes that you can carry into the cells or out of the cells, or into the organelles or outside of the organelles, you have to wait until these proteins empty, so they can carry more solutes. Now the solutes, or the substances that move across the plasma membrane, they are not permeable to this plasma membrane, so that's why you have to use a carrier. There are certain substances that can move freely across the plasma membrane, like CO2 or oxygen. These are gases that are very small, but they can cross without any carriers through the plasma membrane. But certain substances like glucose, which is very large, or other solutes, they have to be transported with the use of these proteins, transport proteins. Now the solute will be also known as the ligand, and this will bind to the receptor sites on the carrier protein, and then the solute will be transported unchanged to the other side of the plasma membrane. And when you saturate all your transport proteins, you will decrease the rate of transport. So this is what we call the transport maximum, which is the transport rate at which all the carriers are occupied. So this graph shows you how you can saturate your carriers. So you start transporting substances very efficiently up to when you have saturated all your carriers with the solute, and then you reach a plateau that is known also as TM, or transport maximum. Now in terms of carrier-mediated transport, so we have three kinds of carrier molecules. So we have some that carry only one type of solutes, that these are called uniports. Example of a uniport will be a calcium pump. You can have some carriers that can carry 2 or more solids at the same time, and this is in the same direction or in opposite direction. So you have symports that carries 2 or more molecules simultaneously in the same direction. So we have, for instance, the sodium glucose transporters that we have in our intestinal cells that can carry a molecule of sodium and a molecule of glucose at the same time. You can have the transport of 2 or more solutes in opposite directions, and this is known as countertransport, and it is given by antiports. Example, we have the sodium-potassium pump that removes sodium and brings in potassium. And the 3 mechanisms of carrier-mediated transport uses energy. So you have a primary active transport and a secondary active transport, and then you have facilitated diffusion. Sorry, facilitated diffusion doesn't use energy, so it doesn't consume ATP. And facilitated diffusion is bringing molecules from one side of the cell into another using these carriers down its concentration gradient until you reach an equilibrium, so from more to less. So the solute in this case will attach to the binding site of the carrier. Then the carrier will sense this physical distortion, it will change its shape, and then will open and then release the solute on the other side of the membrane. So this is for facilitated diffusion. So you have this solute with represented in red spheres that binds to this site for this carrier, and when this site on the carrier grabs this solute, you have a transformation of this carrier, and then this carrier changes its shape, closes in here and opens in here, and then it transports this solute from one side of the cell into the other side of the cell. Now, in primary active transport, the carrier will move solutes through the membrane up to its concentration gradient. So in this case, you have to have the use of ATP because you have to move substances from an area that has less concentration to an area that has more concentration. Examples of this will be the calcium pump, which uses ATP to transport calcium from the cell to where it's already more concentrated. In the case of the antiports, you have sodium-potassium pump that uses ATP while putting sodium outside of the cell and bringing potassium into the cells. Now, the sodium-potassium pump, it is found in many places in our body, and it is using as much ATP that half of our daily calories are used for the functioning of these sodium-potassium pumps. They are especially located in cells that are excitable, like the nerve cells and our muscle cells. So because of the action of this pump, you exchange 3 sodiums out of the cell while you're bringing 2 potassiums into the cells. So this is the dynamic equilibrium or homeostasis that we were talking about. You have changes in concentrations of ions, in this case sodium and potassium, to maintain these nerve cells and then these muscle cells functioning properly, so to maintain homeostasis. And one of the bases for anesthesia is blocking these sodium-potassium pumps in these nerve cells so that you don't feel pain when you have an intervention. So what this sodium-potassium pump does then is to keep the concentration of potassium higher inside the cells, which is already high, and hide the concentration of sodium outside of the cells, where the concentration of sodium is high. So this is how the sodium-potassium pump works. Another name for the sodium-potassium pump is known as sodium-potassium ATPase because it breaks down ATP into ADP, or adenosine phosphate, and inorganic phosphate. So in order to maintain the normal concentration of sodium and normal concentration of potassium, you use this pump. So you have to have high concentration of sodium outside of the cells and a high concentration of potassium inside of the cells. So in order to do this, you will grab with this antiport 3 sodiums on one side and 2 potassium on the other side. Sodium is depicted with orange color and potassium with blue color spheres. So you transport then 3 sodiums out and 2 sodiums in. 2 potassium in. So you maintain this homeostasis. So sodium highly concentrated outside of the cells, while potassium highly concentrated inside of the cell. So it's up to a concentration gradient. So because of this, then you maintain a steep sodium concentration, allowing for secondary active transport. And then this also helps to regulate the cell volume because you prevent the cells from gaining too much water by keeping sodium outside of the cells. If you have high concentration of sodium outside of the cells, the water will follow sodium. And it also helps to maintain a negatively charged resting membrane potential. Resting membrane potential will be the voltage that our cells have when the cells are not active. So if you're kicking the positively charged sodium outside of the cells will have a negative voltage, which regularly they have because many proteins or macromolecules, like in the case of DNA, they have phosphate groups that helps to maintain these negative charges inside the cells. And through this function of breaking down the ATP with this sodium-potassium pump, you release heat. Now for secondary active transport, you will have a carrier that moves the solid through the membrane but only uses ATP indirectly. Example would be the sodium glucose transporter, or SGLT, which is a symport. So in this case, you grab a molecule of glucose and a molecule of sodium at the same time, and you move them down their concentration gradient. So outside in the lumen of our intestinal cells, or intestines, we have, if we have ingested in our diet, a diet that is rich in sodium and glucose, you will gain more glucose coming from the lumen of the intestines into the inside of the cells. And this will depend on the primary transport performed by the sodium-potassium pump, and does not itself use ATP. But since you will be generating at some point transport performed by the sodium-potassium pump, you will use ATP because this sodium-potassium pump will require ATP to kick out the sodium outside of these intestinal cells. So another place where you can find these sodium glucose symports will be in the kidney cells that have also this sodium potassium pump at the other end of the cell and prevents the loss of glucose to urine. So this is how we have this coupling of the sodium glucose cotransporter in the kidney cells and how we have the sodium-potassium pump at the basal side of the cells. So as you are transporting into the cells this sodium and glucose at the same time through this symport, as you can see, glucose is in green and sodium in orange, you increase the concentration of sodium inside the cells. But this sodium cannot be highly concentrated inside the cells. It has to be moved out of the cells. And that's how you use this sodium-potassium pump at the basal side of the cells. And as you are moving sodium out, you are bringing this potassium in, and then you use ATP because this is the sodium-potassium ATPase that uses ATP. Now we have another transport in the cells that is called vesicular transport. This is the movement of large particles or fluid droplets or numerous molecules that pass through the membrane in vesicles, like in little pouches or bubble-like enclosures of the plasma membrane. Now this vesicular transport, when it's bringing molecules into the cells, is called endocytosis. Now when you are moving particles or molecules out of the cells, it's called exocytosis. And you use motor proteins that are energized by ATP. Now in endocytosis, when the cells are engulfing large particles through the use of pseudopods, this is known as phagocytosis. And these vesicles, once are inside of the cells, are going to be called phagosomes. And some cells that are cells that eat up debris are known as the macrophages. Now, if the cell ingests fluid droplets, this process is going to be called pinocytosis. And the membrane will cave in and then pinches off the pinocytic vesicle. Now, in endocytosis, we have something that's called receptor-mediated endocytosis, which the particles will bind to specific receptors of the plasma membrane, like the case of clathrin-coated vesicles. So this is a cycle showing you endocytosis and phagocytosis together. So you start with this cell, and you see the nucleus of the cell. This is a phagocytic cell that encounters a foreign particle. And that foreign particle is going to be introduced into the cells by the production of these pseudopods that are going to embrace this foreign particle, and they are going to form like a little pouch of the plasma membrane that is going to encluster this particle into what we call a phagosome or a vesicle, which is found in here in step 3. And this particle, this phagosome, is going to fuse with a lysosome or a vesicle in the cells that has enzymes. And you're going to form, as you can see here on step 5, a phagolysosome. These enzymes are going to break down the chemical bonds in this foreign particle and it's going to produce pieces. These pieces might not be needed for the cells to use, and the cells are going to get rid of these phagolysosomes. So they're going to move this phagolysosome close to the plasma membrane in the step 7, which is known as exocytosis, in which indigestible residues are going to be extruded from the cells. Now, in receptor-mediated endocytosis, you have a more selective endocytosis or bringing of particles. So this will enable to take specific molecules that binds to extracellular receptors, like in the case of hormones. And then you form a vesicle that it is coated with a protein that is called clathrin, and this will be recycled back into the cytoplasm, so you can bring in more particles. One example for this is the uptake of LDLs or low-density lipoproteins from the bloodstream. So this is the figure showing you the process of receptor-mediated endocytosis. So in this case, the plasma membrane has these protein receptors that binds to these extracellular molecules. These can be signaling molecules or they can be hormones. And this panel up here is the electron microscope images of these receptor-mediated endocytosis. So here you have your receptors. It binds these extracellular molecules. It forms this pinching of the plasma membrane or sinking of the plasma membrane inward, and then you coat it with this protein that is called clathrin, and this vesicle will pinch off from the plasma membrane. Then you use these molecules, and then once these receptors are empty from the molecules, they are returned back into the plasma membrane to catch more extracellular molecules if they are needed. Now, when the cells are transported only across the cells without doing anything to them, this is called transcytosis. So they can enter through the apical side, and then they can be extruded through the basal side or vice versa. In exocytosis, you secrete only the material, and then you replace the plasma membrane that it was previously removed by endocytosis. So in transcytosis, basically you move the vesicles from one side of the cell, which is this, into the other side of the cell. So these are muscle cells, and this is the fluid outside of the muscle cells. Sorry, these are capillary cells, and these are muscle cells on this side. So you have these pinocytic vesicles or fluid-filled vesicles or liquid-filled vesicles that move from the blood that is found in the capillaries into the tissue fluid that it is outside of the muscle cells. And remember again, tissue fluid is also known as interstitial fluid or extracellular fluid. So in this case, they're showing you a process of endocytosis in which you catch these molecules through this plasma membrane, and then you form a little dimple, and then this exocytosis is where you want to extrude these substances that might not be useful for the cells. So you form first a bridge between the plasma membrane and the secretory vesicles through a linking protein, and you form a dimple. And as you're pulling the plasma membrane, this plasma membrane will fuse with the vesicle plasma membrane, and you will form something that's called a fusion pore, and then you secrete the substance. And this is the electron micrograph showing you how is the process of exocytosis. Now, another component of the plasma membrane, we are done with the transport mechanism, it is the cytoskeleton. So the cytoskeleton, it's a network of protein filaments and cylinders that helps to the cell to have a particular shape, to support the shape of the cells or the structure of the cells, to organize the contents of the cells, to direct the movement of materials within the cells, and also to make the cells to move by the creation of pseudopods. The cytoskeleton is made out of three proteins: the thin protein filaments that are known as microfilaments, the intermediate fibers that are made mainly out of keratin, and microtubules that are formed by tubulin proteins. So in this figure, you can see the three cytoskeletal proteins. So this will be the microtubules with this tubulin protein. And then you have here actin filaments, which are also known as microfilaments, forming part of this microvilli. And then these intermediate filaments that are made out of keratin. And then you can see here in these cells, which are more than likely fibroblasts, you can see the nucleus and then these filaments of proteins inside the cells, the cytoskeleton, that are stained with yellow color. So microfilaments, they have a thickness of 6 nanometers. Again, they're made of actin. Intermediate filaments, they're made of keratin, and they have a thickness between 8 to 10 nanometers. Functions of the intermediate filaments is give the cell the shape and resist stress. And the microtubules are the thicker parts of the cytoskeleton, so they measure 25 nanometers in thickness. They are made of tubulin proteins that they radiate, forming a centrosome that can come and go. And some of the functions of the microtubules is to maintain the cell shape, to hold the organelles like even the chromosomes inside our nucleus, and to act as a transport mechanism through which the substances or proteins in our cells can move. And then they form axonems of cilia and flagella, and they can form the mitotic spindle to separate the chromosomes. So this is the structure of the microtubules. So the microtubules are formed of units that are called protofilaments, and if you extend one of these protofilaments, you can see this globular protein that is called tubulin. And attached to some microtubules, you have these dynein arms, and they can form stacks like this. Now for the organelles. So we have membranous and non-membranous organelles. The membranous organelles will be the nucleus, because they have a membrane. That's why they're called membranous. So you have the nucleus, the mitochondria, lysosomes, peroxisomes, endoplasmic reticulum, and Golgi complex. So all of these organelles, they have a membrane. Now the organelles that they don't have a membrane: ribosomes, centrosomes, centrioles, and basal bodies. Now the first membranous organelle, the nucleus, is the largest organelle. It can measure up to 5 micrometers in diameter, and most of the cells in our body have a nucleus, with the exception of the red blood cells, that they don't have a nucleus, so they are anuclear. And some cells, they might have more than one nucleus, and these are multinucleate cells, like in the case of the skeletal muscle cells. Now the membrane of the nucleus is the nuclear envelope. It's a double membrane that surrounds the nucleus, and it has nuclear pores, which are little doors that allows the import and export of molecules. The nuclear envelope is supported by a membrane that is called the nuclear lamina. The nuclear lamina, it is made out of protein filaments, and they provide a point of attachment for DNA or chromatin. And this will help to regulate the cell life cycle. Now, the cytoplasm of the nucleus is known as nucleoplasm. And within this nucleoplasm, you have the chromatin or the DNA, and you have proteins. And also you have a small organelle that is called the nucleoli, which is the place where you make the ribosomes. So this is an electron microscope image of the nucleus. So in this case, this is a cross section of the nucleus. So you have the nucleolus in here. You have the fluid part of the nucleus that is called the nucleoplasm, and in here you have the nuclear envelope. You cannot see the chromatin or the DNA, but it is scattered somehow through here. And then with this freeze fracture of an electron microscope, you can see the characteristics of the nuclear membrane. So this is the nuclear membrane, and then you can see these gaps that are called the nuclear pores, through which substances can move in and out of the nucleus. Now the nucleus, it is connected to the rough endoplasmic reticulum. The rough endoplasmic reticulum is a membranous organelle that has these stacks or sacs, and outside of these sacs you have these ribosomes. Ribosomes are made of ribosomal RNA, and this ribosomal RNA helps to make proteins in the process that we call translation. Now, the endoplasmic reticulum is the generic name for this organelle that has cisternae, or these stacks of membranes that acts like a channel or channels. Now we have two types of endoplasmic reticulum. We have the rough that I already told you about, that is covered with ribosomes. And because of these ribosomes, when you look at this rough endoplasmic reticulum under the electron microscope, you see it like a rough surface. And this rough endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope to receive the messenger RNA that is going to bring a message from the nucleus into the ribosome so that the ribosomes can make certain proteins. Now, another function of the rough endoplasmic reticulum is to produce phospholipids, not only proteins, and these phospholipids and these proteins are going to form part of the plasma membrane. Now, when this rough endoplasmic reticulum is synthesizing proteins, these proteins are packed into other organelles like the Golgi apparatus or Golgi complex, or they can be secreted from the cells. Now we have another type of endoplasmic reticulum that is called smooth endoplasmic reticulum, or SER. This one is called smooth because in its membrane it doesn't have ribosomes, and when you look at it under the electron microscope, you don't see this surface as rough, you see it as smooth. Now the cisternae, or the channels in the smooth endoplasmic reticulum, are more tubular and branching than for the rough endoplasmic reticulum, and the cisternae is thought to be continuous with the rough ER. One of the functions of the smooth ER is to synthesize steroids and other lipids, and also it detoxifies alcohol and other drugs. So this smooth endoplasmic reticulum is more abundant in cells of the body that helps to get rid of alcohol and drugs, like in the case of the liver cells. Also, smooth endoplasmic reticulum serves as a storage for calcium, and rough and smooth ER are functionally different parts of the same network. So you can see here this network of endoplasmic reticulum. The one with ribosomes is the rough endoplasmic reticulum, or RER, and the one without ribosomes is the smooth ER. So the smooth ER doesn't make proteins, but the rough ER makes proteins because it has the ribosomes. Now, what are the ribosomes? They are small granules of protein and RNA. They are found in the nucleoli, that's where they are made, and also in the cytosol. And they are found, of course, attached to the membrane of the rough ER and to the nuclear envelope. So the ribosomes, they read the code sent as messenger RNA. And then it will start assembling amino acids into proteins that are specified by the genetic code. Now, the Golgi complex is a series of cisternae as well, or channels, that synthesizes carbohydrates and attach them into the proteins, and it gives them like a tag to know if the proteins are going to be exported or if the proteins will stay in the cells, and if they stay in the cells, into what part of the cells that they go. So it's like a UPS or a postal service, because it will sort the proteins and it will deliver them into the right area in the cell. what are the tags that they put into the cells? Will be carbohydrate moieties. And some of these proteins that are found in the Golgi complex, they are going to be enzymes. And these enzymes, they will be transported into vesicles that we know as lysosomes. And some vesicles will migrate to the plasma membrane and fuse to it, and some will become secretory vesicles that will store a protein product for later release. So this is how the Golgi complex looks like. It's a series of cisterna or stack of channels, and then these channels will release these vesicles, Golgi vesicles, that they can have enzymes, they can have proteins. And these lysosomes, which are pouches filled with enzymes, are generated within this plasma membrane, within this Golgi apparatus or Golgi complex. And these lysosomes, they have a shape that is generally round, but they can be variable. So functions of the lysosomes is to have enzymes that can digest proteins to hydrolytic process, and also they can help to digest proteins, nucleic acids, complex carbohydrates, phospholipid, and other substances. So for instance, when our cells get damaged and they cannot be repaired, these lysosomes, they release their enzymes, or the organelles that are damaged, they get digested by these lysosome enzymes in what we call autophagy. And then the cells will commit cell suicide into what we call autolysis. Now, peroxisomes are inclusions in the cells that resembles lysosomes, but they have enzymes that are enzymes that neutralize the radical molecules in our cells, the free radicals, the ones that I told you in the previous chapter that they can damage the cells by destroying molecules within the cells. So what is the function of the peroxisomes then? To use molecular oxygen to oxidize it to organic molecules, which are no harmful. So within these peroxisomes, you will have reactions that produce hydrogen peroxide, or H2O, and you have an enzyme that is called catalase or peroxidase that can break down this hydrogen peroxide, which is toxic to our cells, into water molecule, H2O, and into oxygen. So function of these peroxisomes is to neutralize free radicals, also to help to detoxify alcohol, other drugs, and a variety of blood-borne toxins. It helps also to break down the fatty acids into acyl groups for the mitochondria, another organelle, to synthesize ATP. And these peroxisomes can be found in all cells, but they are abundant in cells that get rid of toxins like the liver and the kidneys. So if you look at a microscopic view of these peroxisomes with a transmission electron microscope, you can see these lysosomes at a low magnification and these peroxisomes at a higher magnification. Now, proteasomes are hollow cylindrical organelles that can destroy surplus proteins or proteins that are misfolded or non-functional. So they will have enzymes that can break down proteins that were tagged for destruction. And then you produce short peptides and amino acids that can be recycled to make new proteins. So this is what happened with the proteasomes. So they are cylindrical structures that has enzymes inside that can destroy these misfolded proteins that were tagged for destruction. So you add this tag into this protein that it was misfolded, Then you put it into the proteasome, and then you make amino acids, and then these amino acids, you will use them to make more proteins. Now, mitochondria is a sausage-like organelle. It has the size of a sausage, the shape of a sausage, and this organelle is the powerhouse of the cell because it synthesizes the majority of the ATP. So it can change its shape from sausage to thread-like in some cases, and this organelle, it has a double membrane. And this inner membrane, it falls inside and forming this structure that is called cristae. The spaces between the cristae is known as matrix, and in the matrix is where we have ribosomes and enzymes that are used for the synthesis of ATP. And mitochondria, it's a special organelle because not only produces ATP, but it has its own DNA molecule, which is circular DNA, and is known as mtDNA or mitochondrial DNA. So this is the structure of the mitochondria. This is seen with a scanning electron microscope. And this is, of course, the cartoon. So it has the double membrane, the outer membrane, and the inner membrane. The inner membrane folds in, forming cristae. And then you have ribosomes in here, you have DNA, and you have enzymes that helps to form ATP. Now the centrioles are a couple of cylindrical structures that resemble microtubules because they are arranged in 9 groups of 3 microtubules each. And 2 centrioles will lie perpendicular to each other within the centrosome, which is a small key area of the cell. And these centrioles play an important role in cell division because as the cells are dividing, these centrioles will migrate to opposite poles of the cells, and then they will form a structure together with the microtubules that are going to bind into the center of the chromosomes into what we call the mitotic spindle, and then you separate the chromosomes into 2 daughter cells. Now these centrioles form basal bodies of cilia and flagella. And in each basal body, there is a centriole that originated in central organizing centers that has migrated to the plasma membrane or to the membrane. So if you make a cross section of these centrioles, if you cut it like that, you will be able to see these microtubules arranged in 1, 2, 3, 4, 5, 6, 7, 8, 9 groups of 3. And this is how it looks with a transmission electron microscope. Now the cells not only have organelles, but they also have inclusions which are used for different purposes. So you have inclusions that stores substances that are beneficial for the cells, like glycogen molecules that are rich in glucose, pigments or fat droplets, and also some inclusions they can have foreign bodies like viruses, bacteria, particles of dust, or debris that were phagocytosed by the cells. And the inclusions are never enclosed in a unit membrane, and they are not essential for cell survival. Okay, so this is all for this chapter. This is the end. If you have any questions, please let me know, and I will see you in the next video. Bye-bye.